Histology is the study of tissues, a science that revolves around a single cell. All human cells have much in common, but there are important differences among our cells, and it's upon these differences that histology is founded. The hundreds of specialized cells in your body make up 4 groups of human tissue. The different form and function of these tissues can be as distinct as these nerve and muscle fibers are to sperm cells and a human egg. Consider the elegant simplicity of a single red blood cell, identical to billions in your circulatory system. How different they are to the more complex design and organization of those cells forming your intestinal tract. Even more distinct, bone tissue, which supports your skeleton. Vastly different from the nerve cells that make up your brain and spinal cord. And remember that all these physical distinctions and specialized functions arise from the union of just 2 analogous human cells, a woman's ovum and a single spermatozoa from a male. And then, miraculously, after 2 trillion mitotic divisions occurring over 9 months, we emerge as the most complex biological organism on Earth, awesome in design and potential. Let's now take a separate look at the 4 essential categories of human tissue. We'll begin with epithelial tissue. We'll move on to connective tissue, then we'll examine muscle tissue, and we'll finish with a look at nerve tissue. Epithelial tissue. The characteristics of epithelial tissue include cells that are very tightly joined, often interlocked. Cells that often form or are found on an exposed surface, and cells that are avascular, meaning not pervaded by blood vessels. Epithelium can be classified according to the degree of cell layering, which might be a single layer, multiple layers, or variations thereof. And also based on cell shape, which might be flat or squamous, boxy or cuboidal, or tall or columnar types. Functions of epithelial tissue include providing some degree of physical protection to underlying cells, secretion or absorption, especially along the GI tract, and sensory reception, as we see in the epithelium on the tongue. Structures formed include the surface of the skin, glands that secrete both internally and externally, membranes within the body, and linings of hollow organs. Let's now look first at squamous epithelium, so called because the cells are flat or scaly. And let's look at simple squamous epithelium, where the cells are 1 layer deep. These can be collected out of the inside surface of your mouth, spread onto a glass slide, stained with a drop of methylene blue, and protected with a cover glass. Under low power, they appear flaky and flat, and higher magnification shows how they were once joined, tightly connected in their natural state. In the lab, prepared slides of simple squamous epithelium are often labeled mesothelium, such as the sample seen here. This form of simple squamous lines internal membranes, such as the pericardium around the heart. Simple squamous also lines blood vessels, providing a smooth surface to improve blood flow. Squamous epithelium is more often stratified, meaning multiple layers, sometimes 10 or more cells deep. This layering provides more resistance to mechanical stress. Here's a low-power cross-section at the surface of the skin. The stratification can be appreciated, especially at higher magnification. Notice how the tough and dried cells at the surface appear to be breaking away and replaced by epithelial cells arising from below. This is called exfoliation and is responsible for the renewal of skin, which is constantly exposed to the elements. And here's an electron micrograph showing exfoliation of dead squamous cells. Cuboidal epithelium is typically not stratified, so we'll look only at simple cuboidal epithelium. This epithelium is composed of box-like cells and is frequently seen forming tubules or ducts, as within the kidney. Here at low power, zooming into higher magnification. Simple cuboidal cells are also often secretory and form the internal landscape of many glandular structures throughout the body. Columnar epithelium is also not often stratified. Thus, in our lab, we'll consider just simple columnar epithelium. These epithelia are found abundantly along the GI tract. And most of our slides representing columnar epithelium are actually from the digestive system. In the small intestine, these tall, column-like cells form microscopic projections called villi, which serve to increase surface area for better absorption of dietary nutrients. Some of these epithelial cells are primarily secretory, releasing mucus or digestive enzymes. And like squamous epithelium, these columnar cells are constantly shed and being replaced. This final view shows an electron micrograph of intestinal villi composed of simple columnar epithelium. Transitional epithelium is a specialized form of unique epithelial cells stratified in such a way as to allow an organ to expand or shrink. Transitional epithelium is found only in structures of the urinary system, such as the bladder, seen here in a scanning electron micrograph. Now, we'll progressively magnify a low-power view inside the bladder in an empty or contracted state. The superficial cells are usually rounded, cuboid cells, making the tissue quite distinct from the other types of epithelia. Before leaving epithelium, let's examine skin, the largest human organ, which is at its surface epithelial tissue. But deep to the epithelial layer is the true skin, or dermis, which contains connective tissues, nerve, and even muscle tissue. A defining component of mammalian skin is hair, which is an epithelial product arising from pockets or follicles which extend deep into the dermis. Hair helps conserve heat and augments sensory reception. Cross-sectional slides of skin may reveal follicles lined with epithelium and rooted in dense connective tissue. Often, oil or sebaceous glands can be seen emptying into the follicles, and many follicles are attached to little bundles of smooth muscle, which elevates the hair in response to chilling or fright, creating goosebumps. And at the surface, layers of squamous over more cuboidal epithelium form the epidermis, which clashes constantly with the external environment. Connective tissues create the internal framework of the body. They're microscopically very diverse. The active cells are isolated and widely spaced. And there's an abundance of extracellular matrix consisting of protein fibers and material called ground substance, both of which are produced by connective tissue cells. We'll classify connective tissue into 5 groups. Loose connective tissue, which includes areolar, adipose, and others such as reticular tissue. Dense connective tissue, where the protein fibers may be regularly or irregularly arranged. Cartilages, which include hyaline cartilage, elastic cartilage, and fibrocartilage. Bone, which features a strong and solid matrix. And vascular or fluid connective tissues, which include blood and myeloid connective tissue. Functions of connective tissue are many and varied. They provide physical protection or support, as in the skeleton. They strengthen and hold things together, as with cartilage. They provide for storage of fat and minerals. And they produce and remove all types of blood cells. Bodily structures that are formed from connective tissue include the bones of the skeleton, connective cords known as tendons and ligaments, flexible and resilient cartilage, blood and bone marrow, and soft connective tissues under the skin, so-called subcutaneous tissues. There are 2 types of loose connective tissue we'll consider. First, areolar, composed mostly of a fluid ground substance reinforced with a low-density protein network. Areolar tissue is familiar as the material you see when you peel skin from a raw piece of chicken. Under the microscope at low power, areolar has a fishnet appearance, with the cells scattered amid the fibrous proteins. The strength of this tissue stems from the collagen fibers, seen here as faint, thick threads. The stretchy properties of the tissue are due to the dark, wiry elastin fibers seen here. Highest magnification reveals both of these protein fibers well, plus the cells that produce the fibers called fibroblasts. Adipose is a connective tissue where most of the ground substance is taken over by fat-filled cells. The weight, volume, and properties of this tissue are attributed to the adipose sites, looking like tightly packed marshmallows. Sections of adipose expose crowded, empty-looking cells, with occasional blood vessels penetrating the tissue. Adipose can serve a role as padding or insulation, but its primary function is to store surplus calories. Each cell's nucleus can be seen pushed to the inner edge of its membrane because the entire interior is filled with a large lipid droplet. Denser, more supportive connective tissues include the cartilages, and let's look first at hyaline cartilage. The word hyaline means glassy, and this smooth tissue can be seen around the ends of bones forming joints. All cartilage assumes and holds a distinct shape. The active cells are called chondrocytes and produce and maintain all components of the matrix. In hyaline cartilage, the collagen of the matrix does not stain very well, and so appears as a clear, usually lavender filling. Hyaline cartilage is somewhat flexible, but actually the weakest of the cartilages. The perimeter is defined by a membrane called the perichondrium, seen here. Apart from joints, hyaline cartilage plays a supportive role along the airways of the lungs, including the trachea and the bronchioles. This stiffens these tubes and prevents their collapse with inhalation. Hyaline cartilage also connects ribs to the sternum and provides there some flexibility to the rib cage for optimum breathing. Elastic cartilage is named for its rubbery and flexible character. This cartilage helps to form the shape of the outer ear. and it also stiffens the epiglottis of the larynx. The chondrocytes are abundant and randomly distributed, and a thick perichondrium is typical. The matrix is filled with short, fuzzy, elastin fibers that usually stain a deep purple. These proteins allow the tissue to be distorted without damage. The strongest of the cartilages is fibrocartilage, reinforced with dense bundles of collagen. This cartilage is located in areas of high compression, such as the intervertebral discs found along the spine. Fibrocartilage is often damaged with age and abuse, and like all cartilage, it is slow to repair because of its avascular nature. The prime distinction of this cartilage is the orderly, often linear arrangement of the chondrocytes and the collagen. This orientation of the collagen fibers follows the stress lines of the tissue and so provides greater durability. The parallel pattern of the collagen is indicated here with these green arrows. Bone is a connective tissue with a hardened but still porous and vascular matrix. Bone tissue makes up our skeleton and has a very recognizable appearance under the microscope. Repetitive osteon units consist of bone cells, or osteocytes, arranged in a concentric circular layer around blood vessels in the so-called Haversian canal. In lab, cross-sections of dried bone display this pattern, but the actual osteocytes are gone. Their location is marked by voids called lacunae. Here's a thin section of bone tissue at low magnification. The large black channels are the Haversian canals. At higher power, the circular array of the lacunae can be seen. And at highest magnification, the cracks in the matrix, called canaliculi, can even be detected. Blood is a fluid connective tissue because its matrix consists mainly of water and water-soluble proteins, forming what is known as plasma. Usually blood is examined as a blood smear, often obtained with a finger prick. A drop of blood is transferred to a clean slide, and another slide is used to squeegee the sample over the glass. This colorized scanning electron micrograph shows the preponderance of red blood cells that give blood its color. But in the lab, smears are dried and stained to accentuate the nuclei of the relatively fewer white blood cells. The red cells, or erythrocytes, carry oxygen and carbon dioxide, while the many types of white cells, or leukocytes, provide for defense against infection. Muscle tissue includes those cells that shorten in ways that contribute to movement. General features of muscle include cells that are elongated, often called fibers, and cells that contract along their longitudinal axis. There are three types of muscle tissue: skeletal muscle, also called striated muscle; cardiac, or heart muscle; and smooth, or non-striated muscle. Muscle tissue serves to bring about movement or to provide support to the body framework. Muscle enables circulation of blood and provides a squeezing action which propels material along. Muscle tissue forms voluntary muscles that are attached to the skeleton. Muscle tissue forms the heart itself, and tubular organs such as the urinary bladder, the stomach, and so forth. We'll begin our comparison with skeletal muscle, which normally makes up most of human body weight. The gross anatomy of skeletal muscle is familiar in the form of meat. In a steak, for instance, the skeletal muscle can be seen grouped into distinct bundles. Under the microscope, skeletal muscle fibers can be teased apart which helps to show their multinucleated and striped or striated appearance. In a longitudinal cut, the fibers run parallel, and the many nuclei are noted along the outer edge of each cell. The striations are also prominent. When cut in cross-section, the striations are not visible, just as a zebra's stripes are not seen head-on. But the multiple, darkly stained nuclei are seen in cross-section, which is an identifying feature of skeletal muscle. The least abundant type of muscle is cardiac muscle, found only in the heart. In sharp contrast to skeletal muscle, cardiac cells are often shorter, branched, and usually mononucleated. When stained, they exhibit striations, but also dark cellular connections called intercalated discs. These help to spread contraction from one cell onto another. In cross-section, cardiac muscle cells are distinguished by their large, singular nucleus, very often in the center of a given cell. Smooth muscle is so called due to its lack of visible striations. This muscle tends to form flat sheets arranged circularly or longitudinally in hollow tubular organs. Here, for instance, SM marks the thick layer of smooth muscle around the opening of the urethra. The contraction of this tissue helps move urine to the bladder. In stained cross-section, especially at high power, individual smooth muscle fibers are seen, layered, and packed together with just one nucleus per cell. But to best appreciate the tapered and spindly nature of these cells, smooth muscle can be teased apart. In this way, the mononucleated and non-striated distinctions are also best seen. Our final, highly specialized category of tissue is neural tissue. Nerve tissue is defined by cells that have stellate shapes with long, wiry extensions, and cells capable of generating, conducting, and processing electrical signals. Nerve tissue consists of two cell types: neurons, cells that have electrical properties and produce signals for communication, and neuroglia, which provide a supportive framework for neurons. Functions of this tissue include sensory awareness, movement in conjunction with muscle tissue, higher order perception and comprehension, and the maintenance and protection of neurons. Nerve tissue forms the brain, the spinal cord, and spinal and cranial bundles coursing throughout the body. Our focus for now will be on neurons only. There are over 20 million neurons within the spinal cord alone. The cell bodies of these neurons are concentrated near the core of the cord and form what is called gray matter. The wire-like extensions of neurons run vertically up and down the cord, forming what is called white matter. When you examine a cross-section of a spinal cord, focus on the central gray matter, shaped somewhat like a butterfly. Here you will spot the cell bodies of the neurons. Most of these are motor neurons that have a multipolar, star-like or stellate appearance with a prominent nucleus. Projecting into a typical neuron are many branching dendrites, and extending away, one lengthy process called an axon. Axons are typically insulated with neuroglia that form the so-called myelin sheath. This insulation keeps electrical signals on track. Nerve tissue is very soft, and so can often be prepared as a smear, which pulls the cells apart. When stained, the cell bodies, axons, and dendrites are all distinguished. Associated neuroglia and cell fragments also dot the landscape. The combination of stellate cells with root-like extensions make the identification of nerve tissue relatively easy. And now, before we get started with our laboratory study, let's see how you do with a short review.